The end is near. How's everybody doing? It's a small group. I expect to have a big group here. However, only you guys will get the knowledge. We'll turn the videotape off, and then you'll get all the good stuff, right? So how's studying going? You can tell me what you haven't studied, because I haven't written the exam yet, and that way I can, well, I won't say which direction that might go, but it could get ugly, I suppose. I haven't even started thinking about writing the exam. Typically when I write the final exam, as you saw last term, it's not quite as long as twice as long as a regular exam. So I'm thinking this will probably be about in that category also, 150 to 180 points total. And the format will be the same as you've seen. So it'll have that sort of short true-false section to begin with. It'll have fill in the blank, and it will have matching. That's pretty straightforward, I think. Our exam is on Thursday at 9:30 in the morning, and I'm sure you guys will all be ready for it. Regular classroom. Material, the new material since the last exam, whatever percentage that works out to be. Yeah. I try to keep it proportional to that. So I guess it's roughly a third, yeah. Yeah, I can't think of anything to say about it. Like I said, I haven't even started thinking about it tonight. I'm going to get home. I'm going to start working on writing it. So any thoughts about what I should put into it? It is cumulative. And reminder for all those in TV land that, yes, you do have to have a note card that you get from me. So that's a requirement for the exam. The rules for the note card, good question. So the rules for the note cards, everything has to be handwritten. No photocopied figures, no layers, no anything like that. Everything in your own hands, and as long as it's handwritten, that's basically what I'm after. Yes, put your name on it. And you can do that at the end if you want to. So just write big letters or something like that, whatever works. So I think it's pretty straightforward. As long as you do it before I call time. That's right. I have had people who've lost points. And in fact, I had somebody last term who lost points for writing after the exam was called off at the end. So, yeah. And I don't do that to be mean, but I do it so that it's fair to everybody. Everybody has the same amount of time. So if one person is, taking advantage of that extra time, then that's extra time that any of you could have used who weren't doing that. And to me, that's almost the equivalent of cheating. So everybody should have the same chance to be successful for it. So that's why I do that. And I make sure that you get a note card. That way, you have to turn one in. So I know that you're turning in a note card. As I've said before, is I don't want people passing note cards on to their roommates, not because there's anything secret on them, but because I think the benefit is in making the note card. So if you pass your note card on to your roommate, then the roommate doesn't benefit from the making of the note card, and that's important. All right, so questions? Bromodomains, yes. Okay. So bromodomains, contrary to what I said, are not regions that are recognized. They are regions that are doing the recognizing. So they are specific protein domains that, I meant to mention that in class, so thanks for bringing it up, Eric. Bromodomains are domains of proteins that bind to phosphorylated tyrosines. That's what they are. Okay. I'm sorry? I'm sorry. I'm back in the phosphorylated tyrosine mode. Yes. Acetylated lysines. Sorry. Yeah. This review session is off to a great start so far. I'm still thinking phosphorylated tyrosines from last term, but yes, it is acetylated lysines that bromodomains recognize. Yes. They are on the coactivator. That's correct. And there are many proteins that have bromodomains. Yes?
You mentioned rhodopsin having vitamin A in it.
Yes.
Is that interchangeable with 11-cis-retinol when you use it in that?
Okay. So thanks also for asking that question. So let me just give a couple pieces of nomenclature. So, and I've had several questions about this. Rhodopsin is what you call the protein opsin, which I didn't describe in class, but the protein opsin, when you combine it with vitamin A, you get rhodopsin. All right. I use the term vitamin A and 11-cis-retinal interchangeably because retinal itself is vitamin A. And all of the isomers of retinal are also considered vitamin A's. So when I say vitamin A, it could be 11-cis, it could be the all-trans, it could be any of those. And there are other isomers that are possible. So all of those are considered vitamin A. Okay? Yeah. Yes? Uh-huh. The loose tight open on the ATP synthase you're talking about? And you want me to clarify what they do? Yeah. Okay. So one of the things that you'll see when you look around different books, you'll see slightly different descriptions in terms of what the loose does, what the tight does, and what the open does. And that's because these things are happening. We think of them as three distinct things. Here's loose, here's open, here's tight. And there's transitions between those. And it's in the transitions between those as to where you draw the line, what's loose, what's tight, and what's open. Okay? So for our purposes, I said loose was the configuration that binds to the ADP and PI. Okay? But in order for that to happen, it has to be open, and open has to bind it. So it's transition from open to loose. So for our purposes, I kept it simple. I said loose is what binds it. Okay? I said tight is what makes the ATP. And I said open is what releases the ATP. Now, if you get reading the book or you get reading other things, you'll see that they'll mix those up because it's that transition between those where some of those states are actually occurring. But to keep it simple, that's what I said. And that simple is what I'm after. Make sense? To tight. Open. Yeah, it makes sense because you bind it, you make it, you release it. You bind it, you make it, you release it, right? One of the interesting things about ATP synthase is it actually can run backwards. It actually can burn ATP. So I didn't talk about it in class. And when it burns ATP, it actually can build a proton gradient, which is kind of interesting. It doesn't normally function that way, but it's possible. Yes? Mm-hmm. Uh-huh. Yeah, so that's part of the thing I said you could teach a whole class on. So obviously after transcription has occurred, you have to have repackaging of the histones in order to go back to the original state. That may not happen immediately. I mean, once you've got it opened up, you may have continued transcription of that gene. At some point, that transcription is going to stop unless it's something we call constitutive synthesis. Constitutive synthesis means that it's always going on at some level, and that you would want to be transcriptionally active. All right. So it's going to vary from gene to gene in terms of whether they're constitutive or whether they're regulated at various parts in the cell cycle. But the way in which they go back and they repackage, that's beyond the scope of this class, partly because we haven't talked about how that overall complex is made in the first place. Yes?
Are there topoisomerases and helicases being used during transcription?
Are there topoisomerases and helicases being used during transcription? Yes, there are. Okay. They're not as active as what you see in DNA replication because it's not going on as rapidly as DNA replication is. But there are topoisomerases and a helicase-like activity that helps transcription to occur. Because it doesn't go as fast, the topoisomerases don't have to work quite as fast, obviously, in my system. black there. Now we're just going to password there. Look out there. Anyway, what was I going to say? So they, oh, I know what I was going to say. So the topoisomerases are kind of interesting. So we've thought of the topoisomerase ahead of the replication fork because we've got these strands peeling apart. And these strands are peeling apart in both DNA replication and in transcription. So there has to be a topoisomerase up front in transcription to relieve that tension. Well, because you're relieving tension up here, and you, if you were to put everything back together, then if you have relieved tension up here and the same tension that you had behind to begin with, then you've actually got a change in the topological density of that, right? So if you start, you peel it, you relieve tension, you put it back together, then you've got less twists than you had before. So on the back side of the RNA polymerase, there's also a topoisomerase to put more twists in. So it actually has to happen on both sides, and that's because you've got an open complex that where the strands are actually staying together. And you don't have that when you have replication because you have a fork. Does that make sense? Sort of. Okay. Yes? Okay, so the difference between an ABC cassette type ATP binding and a P-type ATP synthase is, or a P-type ATP binding protein, is structural only. Okay, so you should know that they're different. All right, but there is a functional difference between them. All right, so first of all, you're not going to memorize what the structure is because we haven't talked about the structure, but they do have structural differences between them. The functional difference is that the P-type always has a phosphoaspartate as part of its mechanism. So it will always have a transient formation of a phosphoaspartate during the catalytic mechanism of a P-type ATP synthase, or P-type ATP binding protein. ATP pump is what I should say. Yeah. So the difference is that the functional difference between the two is that the P-type ATP pumps will have a phosphoaspartate as part of their mechanism. It's transient. It doesn't remain as that. But there is a transient intermediate that has a phosphate on aspartate. Mm-hmm. Yes?
The HGPRT mechanism, according to that?
The HGPRT?
Right.
Yeah, so the HGPRT is the hypoxanthine something, I can't remember the full name of the enzyme. But this is an enzyme that's important in the salvage of purines. And the inability of that enzyme to, well, let me pull up the thing here so I can show you at least what I have on the thing. HGPRT was the nucleotide metabolism, wasn't it? Okay, so HGPRT was, nope, that's the wrong one. Here, is that off? There we go. Okay. So the it was less 9 centers, but I thought I had a figure. I guess I don't have a figure showing that. Okay. So what it is it's a relatively minor reaction in the salvage of purine nucleotides. And we've seen other things relative to purine nucleotides like gout, for example, that arises from a deficiency in the ability to metabolize and make handle uric acid in the body. HGPRT enzyme, when it accumulates, when it's not functional, it accumulates a phosphorylated pyrophosphate and urate, which is also a component of gout. But the important thing here is that it has very severe problems in nerve cells. And the severe problems in nerve cells is what give rise to the Lesch-Nyhan syndrome, which is the one where they basically eat their lips, they eat their fingers, they'll eat anything like that. I haven't talked about what the reaction is, though. So, yeah.
Each cone cell has a unique rhodopsin in it.
Does what?
Each cone cell has a unique rhodopsin.
Does each cone cell have a unique rhodopsin? No. You would have one group of reds, one group of greens, one group of blues, but each one would not be unique, no. But
So there's the red and green.
you would have red ones, you would have green ones, and you would have blue ones. That's correct. Yep. So they would be specific, and then they would be wired specifically to specific regions of the brain. Yeah, it's pretty remarkable. Well, no, they are slightly different. That's why they said, maybe I may have not understanding. So the rhodopsins are slightly different. That's why they have slightly different absorption maxima that they absorb light at. So they're very close, but they're not identical.
Is that is the difference between the cone cells? Or are the cone cells themselves also different?
As far as I know, the cone cells aren't fundamentally different. I think that they just have different rhodopsins in them, as far as I know. Yeah. Yes, Eric.
So the SRE pathway,
Mm-hmm.
when the eukaryotic phase goes to the DNA,
Uh-huh.
that transfers.
Okay, so let's go back to that figure that is problem. So let's see, that was in the cholesterol, right? And that was here. And the SREBP pathway is right here. So this is what you're talking about here, okay? So your question was what now?
The gene, the part that actually binds to the DNA,
Mm-hmm.
transcription factor.
So the part that binds to the DNA is a protein. That's correct. Okay. So any protein that binds specifically to DNA for affecting transcription is a transcription factor. This SRED, I think is what it was. This component right here binds to specific sequences that are found in the promoters of genes involved in hormone responses. And that's why it's called the steroid response element. Okay. So that, when it binds to the DNA, will activate those genes. Yeah. Yes, Denise.
Effective cyanide on coupling.
What's that?
Effective cyanide on coupling.
Effective cyanide on coupling. Cyanide per se doesn't have.
Specifically, effective cyanide on oxygen production /
Okay. Well, let's think about what cyanide does first. So cyanide is an inhibitor of complex 4 in the mitochondria. So let's get you a figure for that. All right. Let's see. And in the mitochondria, that would be here. And where's the transport system? Sequence. Okay, that'll do good now. All right, so there's the movement of the electrons through the electron transport system. And cyanide's affecting complex 4 right here. All right? So it's the last carrier in the overall system. Cyanide has no effect on coupling. That is, it doesn't affect coupling. If the mitochondria are starting out coupled, they will remain coupled. If they start out uncoupled, they will remain uncoupled. So it has no effect on that whatsoever. Your question related to oxygen and what cyanide will do with respect to oxygen is cyanide will stop oxygen being used because it's stopping electron transport. So oxygen consumption will go down as cyanide binds to a complex 4. Yes. Azide and carbon monoxide, that's right. Mm-hmm. So they all would have the same effect. They all would stop oxygen consumption. Yes? What's coupling? Yeah, so coupling means, as the word suggests, so coupling means one thing depends on another. That's what coupling is all about. So coupling with respect to respiration relates to what you've seen in the mitochondrion, and that is that in tightly coupled mitochondria, it means that electron transport is necessary for oxidative phosphorylation, and conversely that oxidative phosphorylation is necessary ultimately for electron transport, because if oxidative phosphorylation gets blocked, then the gradient itself gets too large and electron transport will stop. So 99.9% of the time, your mitochondria are coupled, because if they get uncoupled, we saw the problems associated with lack of making ATP and so forth. So coupling is the normal state of affairs. All right? To uncouple mitochondria, we either have to add 2,4-dinitrophenol to mitochondria to basically poke holes in the mitochondrial inner membrane, or we have to have a situation in brown fat cells that we have in our body where we have a protein called uncoupling protein that will do the same thing and allow ions to flow in. Okay. Yes?
So in eukaryotic translation, does the first tRNA bind to the P site using EFTU, or does it have a specific initiation factor like the
So the question was if in the very first placement of the initiator codon in the ribosome in eukaryotic translation, is EFTU used? And the answer is, well, all these have different names, all right? But the equivalent of IF2 is used. So eukaryotes, like prokaryotes, have specific initiation factors that they use to start the process, and they have elongation factors that do elongation. They have termination factors that do that as well. So the equivalent, the eukaryotic equivalent of IF2 is what brings that tRNA into the start codon and finishes the process of initiation. Okay. I think I mentioned, but if I didn't, I'll say it here. That is that IF2 functions just like EFTU does, in that it helps protect the bond between the tRNA and the amino acid, and it helps to ensure that it's found the right start codon in order for everything to go.
So in prokaryotic tRNA, is there different sequences within the binding tRNA?
Basically not. That one I told you was a pretty rare exception. That's not common. It does happen on rare occasions. Yeah. Yeah. Yeah, you know, somebody else in here asked me that same question, and it was a question that I hadn't thought of prior to that point. And so I went back and I did some research on this myself because I was curious about that very question. Where does the energy for the peptide bond formation come from? And the answer to the question, as best I have been able to find, is that there's not a direct link between the hydrolysis of ATP by EFTU and the formation of the peptide bond. I think what's happening is it's indirect because there's no ATP burned to make the peptide bond. So I think what's happening is indirect, and this is, you're not responsible for this, but because it's a very good question, I want to try to answer it. I think what's happening is that the ATP is being burned, and that burning is actually causing a conformational change in the ribosome that's placing the things into closer proximity so that they can react. It's not actually making them react, though. So it's a very good question, and yeah, it's interesting. I've taught this class for almost a dozen years or so now, and I've had two people ask me that question this year. I've never been asked that question before, so I hadn't thought about it myself. But it's a very, very good question. Yes.
So, with respect to translation, there is position of the
Say it again. I'm sorry.
So, an position to
Inosine. Inositol, something else. Yeah.
how does the proofreading that is exact interpretation?
Okay, so his question is when you have inosine in the third position of the anticodon of the messenger RNA, how does proofreading get by that? Well, the only proofreading that's happened, remember the primary proofreading that's happening is in the attachment of the amino acid to the tRNA. All right, so that's the primary proofreading that's done is at that point, not at the base pairing between the messenger RNA and the tRNA. Okay, so that's one consideration. The other is that inosine does form base pairs with all of those bases that are there. So any proofreading mechanism is looking for base pairing. And so since it does form proper base pairs, it's perfectly fine. It's Another reason, and this is also, we talked about this just very briefly when I talked about the damage to DNA. When you put an oxygen onto guanine, you make something that can base pair with A. And the error repair system has some difficulty with that. And so it's thought that that actually is a cause of an awful lot of mutations because it does form actually a stable base pair. It escapes the proofreading, I shouldn't say proofreading, but escapes the repair reading systems pretty well. Yeah. So in the case of inosine, it's forming a very natural sort of base pair. So there's nothing there to indicate that there's something that's not right. Yeah. And if the question was also about how it gets there, that's a chemical modification that occurs on the tRNA. So it's not synthesized in there. In fact, all the modifications you see to tRNAs occur after the tRNA has been made. So it's a chemical modification that makes an inosine be there. That's fine. Yeah, what's up, Ray?
So with the TRP separator,
Mm-hmm.
when it goes from terminator to anti-terminator,
Yes.
how does that process occur?
Okay.
The mRNA
Okay. It's a good question. Let me bring that up for you. So attenuation is, oh bless, I always do that, is a difficult to understand principle until, yeah, and I wish I had the figure here to show you, because there's a very nice figure, I think I mentioned it in class, that, let's see, where's it at? It was it this? Promoters. No, it's not this one. It's the gene expression. Okay. There. Okay, so attenuation. So if we look at this guy, all right? So we've got, we're looking at the messenger RNA that's been made, and you can see that there are two regions here, a region in red and a region in blue, and either of those can form. And the way it looks here, it's like, well, why don't they both form, right? Why doesn't this one form and this one form, okay? Because there's nothing really stopping both of those guys from forming. And the answer to the question is that this isn't really colored ideally. And the reason it's not is about half of this can pair with half of this. And because they can do that, I like to describe this as having regions 1, 2, 3, and 4. All right? So if we think of this as 1, 2, 3, and 4, 1, 2, 3, 4, then the possibilities for pairing is that 1 can pair with 2, 3 can pair with 4, and 2 can pair with 3. Okay? So what happens is if you get pausing, that is the ribosome is sitting here, it's covering up 1. And when it covers up 1, that is it'll extend about into about right there, okay? 2 and 3 can still base pair. And so when 2 and 3 can base pair, they will. So if 2 and 3 base pairs, 3 and 4 can't base pair, and 3 and 4 is the terminator. So by the pairing of 2 and 3, you're stopping the terminator sequence from forming. Okay? Now, if this guy goes through there, what was I going to say? Very rapidly, then what happens is 1 and 2 get covered up so that 2 can't pair with 3. And it's going through rapidly when there's plenty of tryptophan. So it goes through rapidly. The ribosome sits here, and this now allows 3 and 4 to pair. So when 3 and 4 pair, they terminate, and the thing stops right there. Does that make sense? I really should dig up that other figure I've got that shows the 1, 2, 3, 4, because it's really very cool what is in those messenger RNAs. Sure. In fact, I'll tell you what, if I I think I can find it, and you can pull the URL off yourself. How about that? So let me see if I can find it. I used it in my other class. Let's see. It would be OSU 350, and it would be RNA synthesis. Attenuation. There it is. That's the figure. Okay. So there's 1, 2, 3, 4. 1, and now 2 and 3 are paired, which you haven't seen in the other figure. And then there's 3, 4. Okay. So the URL. Oh, this is already a mistake. Plastic. All right. Well, I'll post it. I'll post it. All right. So yeah. That's exactly right. Yep. So the speed is inherent to the overall success of this thing. And speed is not too surprisingly a function of concentration of the individual tRNAs that are there. It's just, it's really interesting how that has happened. Yeah, so his question is whether it's a knob or, is it being turned on or off? And we like to think of it as being, that really on or off thing. Again, it's not. So it's not a perfect system. Is it going to turn off every single time that there's plenty of tryptophan? No. Is it going to sometimes terminate when it's short of tryptophan? Yeah. But overall, it's pretty good about that sensing. So, and the fact that it's evolved for several of the amino acid operons suggests that it's a very functional system. So it seems to work pretty well for cells. So attenuation is just the entire mechanism by which the operon's controlled. So basically what you said, it's the turning on or turning off, yes. Okay? Get back to 450 here. Any other questions about that before I take it away? If I forget, remind me, and I'll send a link to this out to the class. No. And in fact, it wouldn't work in eukaryotes. That'd be a really good exam question. Why wouldn't it work in eukaryotes? You guys should know this one. No, not introns. There's something else that's different in eukaryotes. We don't, but there are eukaryotes who do. No. Think, guys. What has to happen here that doesn't happen in eukaryotes? They don't happen at the same time. So transcription and translation are separated in eukaryotes. So transcription happens in the nucleus, translation happens in the cytoplasm. This requires them both to be happening at the same time in the same place because translation is affecting transcription, right? That would have been a great exam question. I haven't written it yet. So maybe I should think of another hard one like that, huh? You guys are inspiring me here. This is good. No, I didn't say that. You guys said you didn't want to. Yeah. I didn't say I just.
That's an example of the question that we come up with. I don't want.
It's interesting because this is the first class I've ever had say no to that. And every other class that I've asked it, they all overwhelmingly wanted to do it, and they all, I didn't get very many questions, but I did get, a few. And I always used questions, and I thought they were very fair questions. They weren't evil questions, actually. But what this says is you guys don't trust each other. In other words, you trust each other less than any other class I've ever had. Well, this class has been very, very good about asking a lot of questions. Yeah. So maybe that's what it is. Maybe you're right, Andrea. Is that right? Yeah.
Been there, done that.
Yeah. I think that the ones I've used in the past weren't very different from ones I would have used. So I believe it or not, I really don't try to trick you guys. I mean, I really don't try to do that. So it's important that an exam be a fair measure of your knowledge of the subject, and I really want that to be the case. Yes? Phosphatidyl synthesis? Okay. Haven't said too much about that, but I can go through what I did. Let's see, that would be glycerophospholipids. Where were glycerophospholipids? Now I'm drawing a blank here. Here it is, lipid metabolism. Okay. So, there is the one figure that I sort of used for that. So, glycerophospholipid synthesis uses activated intermediate to accomplish what it does. And the activated intermediate is linked to CDP. So cytidine nucleotides play a role in the synthesis of glycerophospholipids. All right? They don't play a role in the synthesis of sphingolipids for the most part. That CDP can be attached to two different things. It can be attached to the phosphate of a diacylglycerol phosphate, DAG, or it can be attached to the molecule that's being attached to diacylglycerol phosphate. All right? So either way, an activated intermediate is used to make that. So if I had a diacylglycerol linked to a CDP and I added serine, then the bond between CDP and the diacylglycerol would be broken, a phosphate would be remaining, and the serine would become attached using the energy of the breaking off of the CMP from that. Okay. Conversely, if I had a CDP serine and I brought it up with a diacylglycerol, then the process would happen with the breaking of the bond between the CDP and the serine. The serine phosphate would get attached to the diacylglycerol, and you would have a phosphatidyl serine. Okay, so it can go either way. And yes, there are very specific ones that go here, don't go here, etc. But again, for simplicity's sake, you get the picture of what's happening. I don't think that memorizing each and every individual one that does or doesn't go that way is very a good use of your time. Yes, sir.
Go over the action of the Terry enzyme.
Which one do we call Terry again? That was
Aminoacyl-Trna Synthetase.
Oh, okay. Aminoacyl-Trna Synthetase. Yes. Okay. So the action of that enzyme. Why do I keep doing that? The action of that enzyme is okay, and the aminoacyl-Trna synthetase. Okay, so that's just the sites. All right. So I'm not sure what to tell you here. What did you have questions about it or what?
I was just unclear on the action.
Well, so the enzyme catalyzes the covalent bond that forms between the amino acid and the relevant tRNA. The Terry enzyme, the aminoacyl-Trna synthetase, has to do two things. One is it has to read the anticodon, and two, it has to attach the amino acid appropriately on the 3' end of the tRNA. Okay? Now, that is the place where proofreading occurs, and that was actually in that figure that I just clicked on. The proofreading occurs within the enzyme where there is a flipping of, here's the activation site, that is the place where the amino acid gets attached, and after it gets attached, it's flipped over into an editing site that determines is the right amino acid on there or not. So this catalysis site obviously should be fairly specific for the amino acid, but it's not absolutely guaranteed it gets the right one. But the editing site ensures that at a reasonable accuracy that the right amino acid is on there. The other part of the enzyme is down here, way down below, and that's touching the anticodon and reading the anticodon at the same time this is happening up here. So that's pretty much what's happening with that. This is a process that takes the equivalent of 2 ATPs. You'll see that there's the amino acid, there's ATP that's being used, and you end up with a MINBO. I just noticed that. End up with an AMP. An AMP is the equivalent of burning 2 ATPs. So it's energetically expensive to put that amino acid on there. As soon as this guy has been made, all right, then that's when EFTU is, very, very tightly associated with that and linked so that it doesn't get exposed to water. And the other thing about this is that the aminoacyl-Trna synthetases, there are, as I said, 2 classes of them. One class puts the amino acid onto the 3' hydroxyl of the tRNA. So remember that a tRNA is a single strand of RNA. It has a 5' end and it has a 3' end. Well, it also has a 2' end because the 2' is right next to the 3, just the carbon number 2 on that terminal sugar. So half of the aminoacyl-Trna synthetases put the amino acid onto the 3' position. Those are the class 2 enzymes. And half put them onto the 2'. That's the class 1 enzymes. And as far as I know, I imagine there probably are people who study it, but as far as I know, the significance of that isn't really understood. You might think about the fact that you've got a 3-dimensional configuration, and so you've got, here's 3' and here's the 2' site. They're right next to each other, but we're thinking about all the important relevance within a ribosome of positioning. And so here we're saying that here's things that could be in this position or this position, and it's still okay to link with this guy up over here. And it just seems a little odd that, to me anyway, that you've got that variable possible position for whatever reason. And I don't know what the reason is. As I said, I'm not sure it's known. So proofreading is actually part of the Terry enzyme. So the Terry enzyme basically proofreads its own work, kind of like DNA polymerase 1 proofreads its own work. Or any other DNA polymerase that does proofreading does that. It's a different kind of proofreading, but yes, it's correcting its own mistakes. Now, there's other kinds of, and I should also make sure that I'm clear about this. There's other kinds of checks that occur to make sure that the proper amino acid is there. This type of proofreading is making sure the proper amino acid is on the proper tRNA. The other type of checking that happens is what happens with EFTU. And EFTU, you may remember, brings in the tRNA. If the right base pairing between the codon and the anticodon is happening, then the EFTU hydrolyzes GTP and leaves. If the wrong one is there, then EFTU pulls that away, and translation waits for another appropriate tRNA to diffuse into the site. Okay. Now, I don't think of that as proofreading because it's not changing something that's there, but it is a kind of a check to make sure that the right amino acid is put in place. Okay. Yes?
The 5S ribosomal RNA.
That's a good question. What is the 5S ribosomal RNA used for? And to be honest with you, I don't know. Most of the RNAs that are in the ribosome are structural domains to actually give the overall body structure. And so I don't know that it has any kind of catalytic function. I'm pretty sure it does not. And it certainly doesn't have anything to do with aligning. So as far as I know, it's probably just part of the scaffolding that the proteins can then bind to and give that overall shape that gives the 3-dimensionality of the ribosome that's needed. But as far as I know, that's the only function. In eukaryotes, you have another small RNA. That's the 5 point. It's actually the 5.8 is equivalent to the 5 of the prokaryotes. Eukaryotes have a 5 of their own that's completely different, and I'm pretty sure in that case it's simply a scaffolding. When I was a graduate student back in the dark ages of the molecular biology, we had to memorize the order in which the ribosomal proteins assembled into making a ribosome. So we had to know they're numbered L1 through whatever 25 or 30 or whatever it is, and then S 1 through 20 or so, corresponding to the large subunit and the small subunit. We had to memorize every one of those, the order that they went together. That was a nightmare. Things you can make graduate students do, you can't make undergrads do. So, howdy. Yes?
Yeah, the iron response binding per element binding protein.
Okay.
It binds both iron and mRNA.
Yes?
Is it binding at the same part of the
Okay.
protein?
So the question is about the iron response element binding protein. Does iron and the RNA both bind at the same site? I would say that they don't bind at the exact same site, but they do overlap. And because they overlap, they can't both bind. So only one or the other can bind. Yep. Yes?
With the aminoacyl-Trna synthetase up here,
Uh-huh.
are there any issues related to putting the amino acid on the wrong hydroxyl?
Question is, if the aminoacyl-Trna synthetase puts the amino acid on the wrong hydroxyl group, are there any issues with that? I don't know, but my suspicion is it would be minimal, because what would happen is you would make one tRNA that would have that, and if you gummed up one protein, it probably wouldn't have any significant impact on the cell. So I don't think that there would be a gigantic impact associated with that. The cell is, as I noted earlier, is willing to accommodate more errors with translation and even with transcription than it is with DNA replication, because both the proteins and the RNAs are transient in nature. They're not around forever. So I don't think there would be a significant problem. You might plug up one ribosome in the process, but there are thousands of ribosomes going on at any given time in the cell. So I don't think there would be any problem associated with that. But I would like to know the answer to the question myself, is are there differences? So you can imagine, I'll tell you what I've thought of with this. So it's one of my pet topics, actually. You could imagine that the rate with which the ribosome moves might be affected by whether it's using something that has a 2 or a 3, because something different has to happen to accommodate a 2 versus the 3. And so it would be interesting to know the three-dimensional configurations of those that have it on a 2 versus a 3, and see maybe there actually is a physical reason why they're in there. So, yeah. There are suggestions that the speed, you've already seen one effect of the speed of the ribosome translating in attenuation, how that can have important implications for the cell. There are a lot of suggestions that the speed of translation itself may be a factor in gene expression. And that's probably true in eukaryotes as well as in prokaryotes. Now, that's not in the same mechanism. It's not an attenuation mechanism. But if the speed is not proper, it might result in lack of production of a protein. So that could be a mechanism for control.
It looks like on the 3D structure of the ribosome with the tRNAs in it, that they were kind of rotating a little bit with it?
Yeah, I don't know about the rotation. I don't know about that. I know what you're talking about, because in the video that we saw, right?
I think it was just a diagram.
Well, there was a video I showed somewhere that actually shows that. You can see a little bit of that movement of the tRNA. I don't know how real that is. Yeah.
Areas of the protein coding that would have greater percentage of class 1 versus class 2 at a speed were really important.
So his question is, if speed were important, would you expect to see a bias for type 2 or type 1?
Yeah. If it
well,
were involved in the mechanism of slowing or speeding up, you would see perhaps a bias.
So my suspicion is that, so I think that's why I'm kind of curious about it, and I just haven't done the research to check it myself. But since you know which ones are type 1s and type 2s, it would be interesting to know if you have a long stretch of 2s and a long stretch of 1s,
Right.
or are they
If you saw that
interspersed?
bias, then it would-
Exactly.
maybe there's something to that.
Yeah, so it could be. Don't know.
I think that'd be relatively easy to check out.
It would, yeah. Time is my only limitation. Did you guys see this thing about, it was on Facebook yesterday, about I love biochemistry? Has anybody seen this? You know what I'm talking about? Nobody's seen this? Oh, goodness. So it's I love biochemistry, right? Except for love is a polypeptide where the first one is the one-letter code for L, the second one is the one-letter code for O, the third one is the one-letter code for V, and the fourth one is the one-letter code for E. It's a beautiful thing, and you look at it, and a person's going to look at it and goes, "What the hell is that?" Okay, only if you know biochemistry and you know the one-letter code does that come across. I thought that was pretty cool. Yes. How does a holiday junction arise? Okay, so a holiday junction arises ultimately because of the sort of strand invasion that I've shown you earlier. So let me get you. Why do I do that every time? So as I go to here and a holiday junction there. Okay, so you see these aligned sequences that are very similar to each other, and you see a nick that has been introduced into this strand and into this strand. This is that strand invasion that I showed you on an earlier figure where you see something like this happening. Okay, so you see this strand, in this case the purple guy, invading this guy over here. If I had made a nick, as was shown in that previous slide, then this red would be invading up in there. All right? So this is only showing you one strand invading, but in fact, both strands will invade if you're undergoing a recombination event. Strand invasion doesn't have to involve recombination, but if we go back then and look at this, we have strand invasion that is getting us to here. So strands invade. And when they invade, what happens is these guys just wind, unwind to let the strand in. And as this one strand here goes in down here, the other strand comes in back in up here. All right. So that is basically a reciprocal event. Now you might think, well, so what? Why is that important? Well, the reason that that's important is when you resolve that, okay, you end up with DNAs that, as you see here, that were not the same DNAs that were there. And this invaded strand, or these invaded strands that you can see right there, are not perfectly base paired. So if you have something that's not perfectly base paired, what's going to happen? It's going to get repaired, and that repair is going to give rise to new sequences, because depending on which way you repair it, you change the sequence of what was ultimately there. So that means, therefore, that recombination will give brand new sequences that didn't exist before. Like red-green color blindness. That's right. Yep. Absolutely. What's that? The what?
I don't want that one. Take it back.
Are you red-green colorblind? Oh.
Just about every other.
Is that right?
Blue and purple is shades.
Huh.
Red and green. I mean, if you get like really bright red, it looks great.
Huh. Well, you should be proud of your uniqueness.
I want to know what people mean when they say they're distinct colors.
Well, I have my own genetic anomaly. I discovered when I went to the doctor a few years ago. I was having abdominal pains, and so they did X-rays and so forth just to make sure there was not some tumor or something that was growing. I mean, there wasn't. But the doctor says, "Oh," he says, "you have is it fifth or sixth sacral vertebrae? I have an extra vertebrae in my sacrum." And yeah, so I said, "Really?" He said, "Yeah." I said, what does that mean? Well, some people have them, most people don't, but it happens during development. And, yeah.
And this occurs, is it equally as likely to reverse and go backwards as to go forward?
Yeah, it's a very good question. His question is, can this thing go backwards as well as go forwards? And the answer is yes, it can. It can back up and go back and leave no change whatsoever. I can't tell you the likelihood of either, but it can reverse and go back and result in an unresolved structure. That's right. It can do that.
So you're a woman, you have both chromosomes, red, green, home cell.
Mm-hmm.
And let's say
Well, okay, so his question is, can a woman have this happen and basically have the problem fix itself, I think is what you're saying. Yeah, the likelihood of recombination in the first place is low enough that a second recombinational event would be so rare that it would be unlikely. Yeah. So I would say the answer to that would be no, for the most part. You guys getting hungry? I know I am. Hungry for knowledge. Well, good. Hungry for knowledge is good. It's low calorie. So I am. That's why I'm very hungry. In fact, you guys are starting to look really tasty to me. I'm thinking of chew on somebody's arm. No, let's see. So far today, I've had yogurt, a little bit of yogurt for breakfast, and I had a pear for lunch. And right now, it's like, argh. Yeah. That's why that's what the whole idea of losing weight is that you don't eat enough to take care of the calories that you're burning. So, yes, I'll eat a decent dinner tonight, though. Are you guys worn out? Yes.
Can you go over the differences between eukaryotic and prokaryotic promoters?
Oh boy, differences between eukaryotic and prokaryotic promoters. I just see there's a bucket of water there. I trust it's water. Okay. Well, difference between eukaryotic and prokaryotic promoters are hard to elaborate, but I'll give you some thoughts. So, prokaryotic promoters are typically bound by one or a few proteins. They're much simpler than eukaryotes, which are bound by many proteins. Prokaryotic promoters are bound directly by, RNA polymerase. And it's the sigma factor of RNA polymerase that helps the RNA polymerase to recognize the sequence and bind to the promoter. In eukaryotes, RNA polymerases don't directly bind the promoter. They're assisted in the binding at the site by all the other proteins that I described to you. Okay. The structure of the promoters are considerably different. So the prokaryotic promoters typically have a -10 sequence that's AT-rich. Eukaryotes also have an AT-rich region called the Tata box that is close to the transcriptional start site, but it can be anywhere from 30 to 100 base pairs away. It's not absolutely required. In eukaryotic cells, it's found only in front of, it's only required in front of promoters that are strong. That is, where there's a lot of transcription going on. It doesn't have to be in front of that. Eukaryotic promoters have enhancers. Prokaryotic promoters don't have enhancers, and that's because eukaryotes need to be able to control tissue-specific expression, and they also have to be able to unwind the chromosomal proteins, as you've seen. So those differences are very, very big. The unwinding of the histone proteins around a transcriptional region is another element of transcription in eukaryotes. It's not in prokaryotes. It's not what I would call the promoter, but it will obviously occur in the region of the promoter. So that's another difference. What else can I think of? Tissue specificity, like I said, enhancers help give tissue specificity. And prokaryotes, you don't have tissues, so you don't have that same consideration. The needs and the demands on a prokaryotic cell are considerably smaller than they are in a eukaryotic cell. Prokaryotic cell has to decide eat, not eat, divide, not divide. That's basically the decisions that a prokaryotic cell has to make. And a eukaryotic cell has to not only eat and make decisions about dividing, but it has to work in harmony with everything else, and it has to be specific for the tissue in which it's located. And so all of those are what make eukaryotic cells much more complicated in terms of their transcription. More than you wanted to know? Or less than you wanted to know, maybe, I don't know. Yes. So his question was that eukaryotes, the enhancer regions can be a long ways away. Does that take into consideration the remodeling that has to happen? And absolutely it does. It does. So the enhancers can help play roles in that process. And so that's why, for example, an enhancer can be located at variable regions away from the transcriptional start site, because once you allow the remodeling engines in to start opening things up, they actually open things up for a pretty wide region. Yeah. So.
That's where looping actually closer to initiation.
That's.
And that's where the role of mediator came in.
So that's exactly right. So his point is that the mediator, which was a protein I said, could actually help to make this loop that happens by bringing together proteins to perform transcription on areas of DNA that are very far away from each other, can work very, very well. That looping actually happens more easily the farther away they're located. If you take a DNA strand and it's only 100 base pairs away, making a loop of 100 base pairs is tough to do. It's just too much constraint, it's too much strain on the DNA to do it. But if you have it a couple thousand base pairs away, it's very easy to make that loop. So it actually works to the eukaryotic cell's advantage to do that a little further away.
Clarify, was aromatase androgens to estrogen only, but not to
Okay, so let's go back to the. So estrogen is a category of hormones. So estradiol being a specific hormone. Okay, and if we go, where is it at? Here. There. Yes. Okay. So these are estrogens down. These are the bottom. The bottom guys are estrogens. All right. So as you can see, in going from testosterone to estradiol, we're making an aromatic ring. That aromatic ring is where aromatase is involved. Now to go from this guy down here, you're also going and making this aromatic ring. So in both cases, the aromatase enzyme is doing that. This was the hormone that several baseball players got in trouble for, right there, okay? Because they were dosing on this in hopes of making this. Yeah. And for a long time, this was actually legal in Major League Baseball. They could inject themselves with this hormone right here, which I think is outrageous. Down here, start growing breasts or something, right? The amount of aromatase would be the only thing. Yeah. 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 It may very well do that. Yeah. Look at those great pecs, right? So. All right, guys, should we call it an evening? One more quick question. That's always a good way to get a quick question. Well, the more aromatase you have, the more of this you're going to make, right? So women will have a lot more aromatase than men will. All right. So I will get this posted probably this evening, but it may not be till tomorrow. I will get it out as soon as I can. And I'll make the exam as hard as I can, too. So, flip side, right?